Hi there, I'm Mrs. Gay. We're still on Chapter 4, looking at Section 4.3. Right? So, as a reminder globally, not just for this test, in this textbook, prokaryotic cells, information about bacteria, is sprinkled throughout the book. And if you remember, when we were sort of talking about this section, I encouraged you to keep somewhere in your binder a little running list of sections in your book that deal with prokaryotes. So if you haven't started one, this would be an opportune time. And so section 4.3 is really our first section. So it's reminding you that bacteria and archaea are the two domains that are prokaryotic. The other domain, eukarya, consists of all eukaryotic cells, and that includes the kingdoms protist, fungi, plant, and animals. Okay? Now, all cells, regardless of whether you're prokaryotic or eukaryotic, have a plasma membrane because you need boundaries. You need to keep the insides different than the outsides. So regardless of your cell type, prokaryotic or eukaryotic, you have a plasma membrane. You also have to have chromosomes because you need information.
So
and all cells have chromosomes, regardless of whether they're prokaryotic or eukaryotic, because they need directions to make things like proteins. Okay? No directions, no exchange of information. That cell's pretty worthless. So all cells are going to have chromosomes. The other thing that all cells are going to have are ribosomes. Okay? Why do you need a ribosome? Do you need to make proteins from the information contained in the chromosome? Okay? So you're going to have those kinds of things. Now, the difference is, if you are, I'm thinking about chromosomes again, if you are prokaryotic, you get one large circular chromosome. You may also have some smaller bits of looped DNA that are called plasmids, which we'll get to in a few chapters. So that's sort of a preview of coming attractions. Okay? If you are eukaryotic, your chromosomes are linear. And if you're eukaryotic, your chromosomes are contained in a membrane-bound nucleus, which is the primary distinguish.
Wait, you mean
eukaryotic? Did I say prokaryotic?
No. Oh, I thought.
Okay. So eukaryotic cells, linear chromosomes, enclosed in a membrane-bound nucleus. All cells, again, have cytoplasm. Now, in a eukaryotic cell, we don't refer to cytoplasm inside the nucleus. We consider cytoplasm the area from the nucleus to the plasma membrane that houses all the other organelles. Okay? Eukaryotic cells are bigger than prokaryotic cells. You saw in the diagram in 4.2. Okay? Prokaryotic cells are only about 1/10 as big as a eukaryotic cell. Okay? And so if we look at this section, prokaryotic cells also have a few special distinguishing features. Okay? Many of them have, so we'll start in the center, you see the nucleus. There's also a nucleoid. The nucleoid is not actually a thing, but rather it's a region. It's the part of the cell where the DNA is located, but it's not enclosed by a membrane. There's ribosomes near the chromosome for making proteins, the plasma membrane, of course. Most bacteria also have a cell wall. Now, the cell wall is made out of peptidoglycan, which is different than the cellulose that makes up plant cell walls, or the chitin that makes up the cell walls of fungi. So many different kingdoms have cell walls, but they're made out of different things. This is why antibiotics kill bacteria, but not us, and not plants. Because what it does is it targets the peptidoglycan, blows holes in it, and then the cell leaks.
Should
Okay?
we know what that cell wall is made up of? Should we know the peptidoglycan?
Again, that's sort of one of those things you should be familiar with, recognize, but not necessarily memorize.
Okay, can you say it one more time?
Peptido, which sounds like peptide.
Okay.
Glycan, which sounds like sugar, right? Okay. Now, we also have many prokaryotes. They have their plasma membrane on the inside. They have a cell wall. Many of them also have a capsule, which is like a jelly-like coating. It helps them stick to things, which is particularly important if you're in a liquid-type environment. Okay. Many of them have flagella, which helps them move through their environment. They also may have pili, tiny little hair-like structures on the outside that help them attach. Okay. But we'll talk, and then, so that's sort of like the introduction to prokaryotes. When we get ultimately to chapter 16, we'll talk about them a lot more. Okay. In section 4.4 on page 56 and page 57, we are going to spend the rest of this chapter considering eukaryotic cells and looking at the organelles. In this case, they're introducing you to the main differences between plant and animal cells. And so, if you look at the animal cell, they have lysosomes and centrioles. And plants do not. Well, a few plants do, but most plants do not. Okay? And if you look over on page 57, you'll notice that there's, on the left-hand side of the diagram, you can see that there are some organelles that are in plant cells that are not in animal cells. That large central vacuole, chloroplasts, cell walls, and plasmodesmata.
Are there any, there's no vacuoles in animal cells though, right?
They're not called vacuoles, we tend to call them vesicles if they're in animal cells.
Or are they the same thing?
They can be used for storage. Mm-hmm. Notice that they both have peroxisomes. Notice that they both have mitochondria. That's key. They both have Golgi apparatus. They both have ribosomes. They both have endoplasmic reticulum, smooth and rough. They both have a nucleus, which is composed of the nuclear envelope, the chromosomes, the nucleolus. And they both have cytoskeletons made out of microtubules, intermediate filaments, and microfilaments. Okay? So, just understand maybe some of the basic differences. They might give you a list of organelles. I could imagine you seeing a list of 5 different organelles, A through E or A through D.
Which one is not
And of that list, they might have 3 or 4 questions associated. So your answer choices, I'm totally making this up. A, nucleus. B, mitochondria. C, vacuole. And C, centriole. And question 2 might be found in animal cells but not in plant cells. Question 3 might be, the produces energy for the cell. And so then you would choose from that, you would use that same set of answer prompts over and over and over again. Answers could be used once, more than once, or not at all. So I could see a listing like that that would ask you to know the differences between plant and animal cells. Does that make sense? Okay. So I'm going to stop this recording now, and I'll answer questions, and then we'll carry on with the next one.